Hi everyone. I'm excited to introduce our fireside chat today on spatial biology and the technology behind it. My name is Christine Shea. I am a board member at Health Tech Build, as well as the Chief Strategy and Research Officer of Day-to-Day Health at Babylon Health. At Health Tech Build, our mission is to accelerate state-of-the-art digital tech within the life science and healthcare ecosystem. So we've had a lot of great organizations speak as part of our events, including many in digital health, as well as digital therapeutics, all the way from smaller companies to large companies, and including large technology companies. And we really look forward to having more organizations participate in this really thriving ecosystem. And so today we have two speakers and a fireside chat. And I'd like to thank our sponsor, MedAcuity, for giving us their resources to host this great event. And I'm going to hand it over to Craig.
Thanks, Christine. I appreciate it. Thanks to everybody for joining us today. I'm Craig Steger, the host of the Fireside Chat, and I'm joined today with Jacques Corriveau, the CEO from UltraView, to join us and talk about spatial biology. Jacques?
Hi, Craig. Thank you. Thank you for inviting me. I'm very much looking forward to the conversation today and telling the audience a little bit about spatial biology and you know, from a larger perspective, but also where UltraView fits into that.
Yeah, I find it fascinating. So I often say a recovering scientist spent lots of time looking at slides and through microscopes and where the technology has really evolved. Why do you think it's taken off so fast in the last couple of years? Or is that just my perception?
No, it certainly has taken off. I think that for me, we really focus in cancer profiling, and so in the cancer world, the success of some of these immunotherapies that have been coming online, where pharmaceutical companies are able to develop therapeutics that allow the immune system to better fight cancer directly rather than traditional chemotherapy. And with the rise of these new therapeutics, it's become increasingly important to understand what is happening in that tumor microenvironment. So what is the interplay between the immune cells and the cancer cells? Is the immune system recognizing the presence of cancer cells? Is it able to and actively trying to attack those cancer cells, sort of zap them, if you will? Or is the immune system completely blind to the presence of the cancer cells? And all of these pieces of information can help guide pharmaceutical companies into determining who's going to be a good responder for a drug and who is not. They can use that information to select the right groups of patients for clinical trials. And ultimately, this type of technology can help physicians determine what therapy is potentially the best fit for their patient, because you know, chemotherapy and other types of cancer therapeutics are very harsh on the human body. And why put a patient through the process if it's not going to be effective in treating them? So this is good for patients. It's good for the healthcare system, and it's good for payers as well.
One of the things that I find fascinating about spatial biology in particular is that it's a nice combination of both, I think, an evolution of technology, because there's a lot of AI and algorithms that are now part of the technology, or of the technique rather. But also it's an evolution, I think, a little bit of the biology and some of the tools we use in the laboratory, and how we're able to, I think, much more efficiently use those. And so in any of those situations, I find it's sort of a chicken and the egg, right? Of which one's more important, sort of thing, and which one's kind of lifting it up. What are your thoughts?
Yeah, it is fascinating. And From that perspective, spatial biology is not new. We've been staining tissue and cells for 150 plus years. Even today, we still use a method called H&E staining, which was a textile stain originally back in the 1800s, and just so happens to stain mammalian cells very, very well. So using that information, if you apply the H&E staining technique to a cancer biopsy, you're able to look under a microscope and see, based on morphology, the shape of the different cells, which cells are immune cells, which cells are oddly shaped and therefore likely cancer cells. And so they've been doing this for, as I said, 100 plus years of trying to understand that interplay in the tissue. And modern day pathologists still do this to determine if a biopsy is malignant or not. I think where it's gotten really exciting, though, is as technology has advanced, we've been able to take that spatial biology that has been run for 100 plus years and get down to subcellular level of detail. So it's a bit like trying to watch a show on a 1970s black and white television compared to a modern HD 8K television. You can pick out a lot more detail. You can understand a lot more of what's happening with that interplay of the cells. You can tell the difference between T cells and B cells, for example, which is very, very difficult to do just looking at morphology or that H&E stain. And then you can combine multiple different omics. So at UltraView, we focus primarily on staining for protein markers, because protein is the actionable molecule for most of these therapeutics. But you can also combine in RNA, gene expression details. You can combine in some cases, mutation detection. And when you combine all of these different omics, you're able to then find patterns that potentially can be used to determine if a patient is going to be a good responder to the drug or not.
Yeah, and where I see the continued evolution. So here at Outcome Capital, I focus on life science tools and diagnostics, and I see this as being like the perfect space, essentially, for where spatial biology as a tool is now starting to have the practical application in digital pathology. And really kind of helping that pathologist move forward, because quite frankly, we're not making as many pathologists. There's not as many of them who want to sit there and look through microscopes, but we can also make them smarter, quite honestly, with the AI sort of component of it. So I do find that sort of evolution fascinating. What do you think is maybe like the next big challenge for the area, for the space?
Yeah, as you can imagine, there's a lot of information to be gathered from the tumor microenvironment. You're talking about millions of cells, various types of cells, and then you've got intracellular material in regions that you're looking at as well. The human eye can really only capture a small amount of that information, and when you're looking at an entire biopsy, you want to be able to capture all the information that you possibly can from that entire tissue section, because once the tissue's gone, it's gone. Nobody wants to have to go back to a patient and ask for another biopsy sample, or maybe it was completely removed and there's nothing to go back to. So you really need to be very careful with that precious tissue sample and keep the patient in mind. As tools advance, you touched on digital pathology, and digital pathology is really about using some of the advanced technologies that we have available to us today, such as machine learning and artificial intelligence, to be able to be trained to find these patterns on their own, because a software tool is able to look at the entire tissue and is able to provide information on what's happening in that tissue, what cells are present, is the immune system recognizing the cancer cells, and then from there, if you have outcome data for these patients, you can compare the patterns that tool is seeing with the outcome data. And if you have enough of this data, you're able to then pull those patterns out that can be applied to prospective patients in the future.
Yeah, it's fascinating how it's going to, spatial biology as it continues to evolve, how it will eventually impact the clinics, sort of thing. And we're already starting to see that in the marketplace. With any emerging sort of technology or technique, essentially in biology, sort of thing, there's obviously the natural sort of ebbs and flows. What do you see are going to be the challenges maybe in the next couple of years for spatial biology to kind of keep on the tear that it's on?
Yeah, I think the biggest challenge is taking these giant data sets and sort of whittling it down to what is truly biologically relevant. In spatial biology, there's a bit of a war happening right now among the technology companies to see how much information they can get out of their samples. So it's a battle of, we can do 40-plex biomarkers. We can do 40 at the same time. You have others that will look at 1,000 genes and give you the gene expression for all of that data. But the reality is that the majority of that data is going to be irrelevant. It's not going to have a pattern to it. It's not going to correlate between people that should be responding, and yet the patterns aren't matching up. It's not making sense. So, what we do here at UltraView is we work with companies that have these large data sets from these very high multiplexing experiments. It's more of a discovery, sort of casting a wide net to see what you can get. But then we'll take those large data sets and we do what we call deplexing, where we're able to whittle down those data sets into what does appear to be biologically relevant. And then we'll partner with companies to build assays to just focus on a defined list of markers on a large sample set. So it lends itself for a clinical trial and clinical trial research, as opposed to basic research that happens upstream and is very important, but that's more in the discovery part of the process.
So actually, so it's really interesting. So explain to us a little bit more about UltraView and kind of how you guys are focusing, because you're from my understanding, you're platform agnostic, and you really have both kind of a services and a reagents sort of play. How have you specifically positioned yourself into this market?
Yeah, thanks for asking that question. UltraView is pretty unique in this space because most companies that are in spatial biology and for that matter, most companies in the life science tools space have a razor blade type of model where they'll sell you a piece of capital equipment that's designed to run their assay and their assay alone. And then they look to sell a lot of consumables on those instruments. We took a different approach. There are a lot of fluorescent scanners on the market. Essentially, any pathology lab has access to these tools and these auto stainers. And we thought, why not take advantage of that large install base rather than selling our own capital equipment, which requires the need to support and service and so on. We started developing assays that are compatible with the 2,000 to 3,000 instruments that are already installed on the market, and that way we can focus just on the science. We can focus on partnering with our customers to develop assays that are biologically meaningful, and then they can run these on whatever fluorescent scanner is available to them. And this is a model that our customers have really enjoyed because they don't have to go through the process of getting budget for capital equipment expenditure. And what if the projects change down the road and they have a $250,000 piece of equipment sitting on their bench that is now not being used. So it's great from our perspective, and I think that as far as service and kits, UltraView does have a CLIA lab. So when you're in the clinical space, clinical research, clinical projects, oftentimes pharmaceutical companies want to outsource that work. They just don't have the bandwidth. It's a smaller team. And so we will design these assays for customers with the markers that they want to look at. And then they will send their samples to us, and we will run those under CLIA conditions to make sure that the quality control is the best that it can be. We do have other customers that prefer to run samples in their own lab. They have plenty of staff to do that. And so we're able to put our product into a kit format and send that out to them. And this also allows UltraView to partner with clinical research organizations like Covance and NeoGenomics and Discovery Life Science and so on, by essentially allowing them to run UltraView assays as well. So they will partner with their pharmaceutical customers, and we will build the kits and ship it to them, and then they can run it at Covance or NeoGenomics.
So for UltraView, on the reagent side of the business sort of thing, it seems like most of the reagents are still all custom. Very few off the shelf, if you will, plex assays. Is that correct?
Yeah, we do have fixed format kits, and we have customers that really like the content of these kits, and it's just easy to buy something that's off the shelf. But the majority of our business is custom, and I think it's just the nature of life sciences and the fact that one researcher is interested in these 4 or 5 markers, and somebody else likes 4 out of the 5 of those and wants to change the 5th, and we offer that flexibility to be able to do that. It's one of the reasons customers love working with us, because we can change those cocktails on the fly in a matter of a couple of weeks and keep the customer up and running.
Do you find as the technique and the technologies continue to evolve, it seems natural in pharma, right, that you move it from the bench to maybe the preclinical animal studies to eventually the clinical studies. Do you see these kits potentially moving towards and eventually getting to being used in the clinic?
We do. We absolutely do. The information that you're able to gather from these assays, it can be very, very powerful, and that's really where UltraView is focused. Our strategy is really to start early in the preclinical process with our customers and establish trust and credibility with them so that they're willing to bring us on that journey. The goal for our customers is to move from that preclinical setting to phase 1 trial and then phase 2 and so on. And at some point, the work we do together does lend itself to signature development, and we are able to find patterns among those patients in those clinical trials that can then be deployed in a later phase trial, and the correct patients can be selected for FDA submission. Before we had these tools, many drugs would not clear the FDA because the patient selection was so random that you really weren't sure how these patients were going to respond. You did your best to try to select, but you didn't have the molecular phenotype for each patient to really include in your trial and submit to the FDA. So now, using these techniques, we can, at a molecular level, determine which patients are likely to be the best responders and only include those patients in the trial. Now, the opposite side of the coin of this approach is that the FDA will clear the drug, but they're also going to clear that signature. So that signature becomes your companion diagnostic assay. So now for a physician to prescribe the drug, they have to have the patient sample run with that same diagnostic test to determine if they're going to be a responder or not, and then they can be cleared to prescribe the therapeutic. So it's it is a double-edged sword a bit, because you want to be able to prescribe the therapies to as many patients as possible, but at the same time, you want to make sure you prescribe it to the ones that are going to have the best chance of success.
Well, and for AltaView in particular, so it also plays nicely into your strategy of working and being essentially platform agnostic. So whether it's a Ventana auto scanner or some of the newcomers that are out there like Luna4, who's looking to do plexes, as they move their platforms or Sekisui's platform or whomever, as they move their platforms into the clinic or they're already established in the clinic, your reagents should be transferable. Is that the idea and that the plan?
Exactly. That's exactly the plan. Our goal is to make it really easy to work with our assays, our products. And so if you already have experience and comfort level with a Ventana stainer or a Leica stainer, if you already have experience with a Zeiss scanner or Polaris or whatever's in your lab, our reagents should be plug and play to be able to run on those. And the folks in the lab are very busy, and if we can save them a headache, we're happy to do that.
So right now, I think spatial biology in particular has more of a preclinical focus. And what we're talking about is sort of, I guess, the evolution sort of thing. So I'm guessing that, and again, I don't know this, but most of your clients seem to be like biotech, pharma, and they're trying to push this evolution. Do you see the market becoming a little bit more of a push-pull sort of thing, where they're pulling it along, but you're also pushing it towards the diagnostic sort of side of the business?
Yeah, absolutely. I mean, we would love to be partners with pharma companies on the companion diagnostic side and help them speed their new therapies through the process and through FDA approval. I'm a firm believer in the fact that companion diagnostic development happens at the speed of science. And so there's very little that we can do, I believe, to force a companion diagnostic through the process. I think that the best that we can do is be good partners to our customers, build the best quality product that we possibly can for them. And these types of steps are going to speed the process for our customers. They're going to be able to get answers on what's happening in their samples, what's happening in the cohort of samples, and trust the data so that they can move to the next phase of trials. And I do believe that with this strategy and the customer base that we have, the strong feedback we get from our customers, that we are going to see these assays in the diagnostic world sooner rather than later.
So it's interesting because you said something earlier that I don't mean to jump back in time, but you said something earlier about the different omics approaches, and it also, some people want to take a different level of plexing. Mm-hmm. But once you bring in the world of diagnostics, there's only so many of a plex that you really can, I shouldn't say that you can do, but that most people are doing now. Is that how you guys have kind of tried to focus, and is that why you guys are there? And can you explain a little bit why you only want to plex so many?
Yeah, that's right. Essentially now the only biomarker that is approved by the FDA for immunotherapies is a single marker assay for PD-L1. And so, I mean, is your strong indication right there that the FDA is very concerned about these high multiplexing data sets, because it's very difficult to analyze all that information. I mentioned earlier that a tumor microenvironment has millions and millions of cells. And then on top of that, you're combining in, 1,000 markers, even 40 or 50 markers. The amount of data, the amount of information you're pulling out of that is so extreme that it's virtually impossible to prove to the FDA that the answer you're giving them is correct. And even if it is correct, how do you go back and show it and prove that? So the important thing is to really whittle this down to the smallest number of markers that seem to be biologically relevant and are correlating to a pattern between responders and non-responders. And the smallest number of markers that you can have in your diagnostic and submit to the FDA, the stronger, the higher probability you will have of clearing the FDA. So, and that's really where we partner. There's a lot of great technologies out there in spatial biology that live in the discovery world, where they're casting a wide net, and they're trying to see what the landscape looks like for expression of these different markers. But from there, you really need to be able to pare that down to the fewest amount that are going to give you the strongest amount of data, and that's what you move forward into your clinical trials with.
So what do you think is maybe the next big challenge for you guys, and for the market in a whole, spatial biology, but then also specifically for AltaView? What do you think is the next big challenge in the next 2 to 3 years?
I think for AltaView, we're always trying to think of new ways to advance the market, new ways to make it easier for our customers to take advantage of the power of this type of technology and information. So here we're really focused on what we call digital health. It's essentially looking at outcome data from patients and comparing that to certain panels that we've created. So for example, we have an 8-plex panel that we call our Immuno-Oncology 8. And it's 8 of the most common biomarkers. And right now we're working to collect patient samples from some of our academic partners and compare the results of that assay to the outcome that this patient had. So what type of cancer did they have? What were they treated with? Did they go into remission? Did they relapse? If they passed away, after how long? And what was their age, comorbidities, all of these things. And when you take that information, you compare it to the profile they got from the kits that we ran on them, you're able to build these large data sets that can be used for downstream data mining. You can license this type of information to pharma companies for their own data mining. And from this information, you can potentially launch and develop your own diagnostic assays. So this is what we refer to as digital health. And then we're also looking at ways that we can bridge the gap between traditional pathology, anatomical pathology, and some of these new advanced techniques. One of the great things about AltaView is that our assay is very gentle on the biopsy samples on that tissue. And so we're able to do the fluorescent staining with our assay, and then also do that traditional H&E stain that I talked about earlier, and overlap the images. So now you get the best of both worlds. You can look at protein expression data at a subcellular level, but also you can see the morphology of the different cells. And what we'd really like to do, and what we're working on actively now, is being able to use machine learning and artificial intelligence to train algorithms with all of that information, so that at some point down the road, a clinician, a pathologist in a lab, in a hospital lab, can take a tissue, stain it with H&E, feed it into a scanner and our algorithm. And based on all the information that the algorithm was trained with, it can make a call just on that original H&E stained tissue. So, when I talk about bridging the gap, it's a way of making it extremely easy for the person in the lab to stain the sample, but the analysis that they're getting from this is much more advanced than what they would have just by looking at it under a microscope.
It's exciting times for the spatial biology world, that evolution into digital pathology and AltaView. So I appreciate your time, and thanks for participating. I want to also thank MediCuity and Health Tech Build for allowing us to have this fireside chat, and hope you all enjoyed it. Thank you very much, and have a good day.
Thank you.